Assalamu alaikum dear students. Today in this video tutorial we will discuss MDCAT test 2025 biology MCQs part 2nd. Dear students, अभी तक अगर आपने मेरा YouTube चैनल को सब्सक्राइब नहीं किया तो अभी सब्सक्राइब कर लें, लाइक और शेयर ज़रूर कर लें, शुक्रिया. If you need MCQs for MDCAT test preparation, let me know in the comment section. The synthesis of RNA and DNA template is known as option A replication, B translation, C transcription, and option D mutation. So the correct option is C transcription. Transcription is the first step of gene expression, where a segment of DNA is copied into RNA by the enzyme RNA polymerase. This process is distinct from replication, which is the copying of DNA to make more DNA. The translation is basically the synthesis of protein from messenger RNA template. In mammals, the synthesis of mRNA is catalyzed by option A RNA polymerase 1, option B RNA polymerase 2, option C RNA polymerase 3, and option D RNA polymerase 4. So the correct option is B RNA polymerase 2. Explanation is RNA polymerase 2 is the primary enzyme for transcribing all protein coding genes in eukaryotes. The RNA it produces a heterogeneous nuclear RNA, hnRNA, which is processed into mature messenger RNA before being translated into proteins. Dear students, subscribe now and press the bell icon. Never miss an update from a technologist. So this is very important MCQs. Introns are present in the DNA of dash. Option A viruses, B bacteria, C human, and option D all of these. So the correct option is C human. Introns are non-coding segment or region of the gene that removed during RNA processing. They are characteristic of eukaryotic genes, which include humans, but generally absent in the DNA of most prokaryotes like bacteria and many viruses. Some DNA is present in the mitochondria of option A prokaryotes, B eukaryotes, C both A and B, and option D none of these. So the correct option is B eukaryotes. Eukaryote cells, unlike prokaryotes, contain organelles like mitochondria and chloroplast. These organelles have their own small circular DNA molecule known as mitochondrial DNA, which encode some of the proteins needed for their function. In RNA, the complementary base of adenine is option A cytosine, B guanine, C thymine, and option D uracil. So the correct option is D uracil. In nucleic acid, specific base pairing occurs. In DNA, adenine pair with thymine, and RNA, thymine is replaced by uracil. So adenine pair with uracil in RNA. Mutations can be caused by option A ultraviolet radiation, UV rays, B ionizing radiation, C alkylating agents, and option D all of these. So the correct option is D all of these. Mutagens are agents that cause mutations. UV radiation ionizing radiation and alkylating agents, as well as known type of mutagens. So, ultraviolet radiation causes thymine dimers. Ionizing radiation creates free radicals that damage DNA, and alkylating agent add alkyl groups to bases, altering their pairing properties. So this is very important MCQs. Substitution of an adenine base by guanine and DNA is known as option A transposition, B transition, C transversion, and option D frame shift mutation. So the correct option is B transition. A transition mutation involves the substitution of purine base adenine and guanine for another purine or pyrimidine base cytosine or thymine for another pyrimidine, since adenine and guanine are both purines. This is transition. Substitution of thymine base by adenine and DNA is known as transposition, transition, transversion, and frame shift mutation. So the correct option is C, transversion. The transversion mutation involves the substitution of purine base for pyrimidine base or vice versa. This case, pyrimidine thymine is replaced by purine adenine, which is transversion. A point mutation results from option A, substitution of a base, B, insertion of a base, C, deletion of a base, and option D, all of these. So the correct option is D, all of these. So explanation is a point mutation is change in a single nucleotide base. This can occur through the substitution of one base for another, or the insertion or deletion of a single base pair. All these single base changes are considered point mutations. Substitution of a base can result in a dash. Option A, silent mutation, B, missense mutation, C, nonsense mutation, and option D, all of these. Correct option is D, all of these. A silent mutation occurs when new codon codes for same amino acids. A missense mutation occurs when new codon codes for a different amino acid. A nonsense mutation occurs when new codon is a stop codon, resulting in the premature termination of the protein. A silent mutation is most likely to result from option A, substitution of the first base of the codon, B, substitution of the third base of a codon, C, conversion of a nonsense codon into a sense codon, and option D, conversion of sense codon into nonsense codon. So the correct option is B, substitution of the third base of a codon. Explanation is the genetic code of degenerate mean that most amino acids are encoded by more than one codon. Often that the first two bases of codon are the most critical, and changes in third base, the uval position, are less likely to alter the amino acid that is specified, leading to a silent mutation. Dear students, if you like this video, please subscribe my YouTube channel. अगर आपको ये वीडियो पसंद है, बराए मेहरबानी मेरे YouTube चैनल को सब्सक्राइब, लाइक और शेयर ज़रूर कर लें। शुक्रिया। So hemoglobin S is an example of option A, silent mutation, B, acceptable missense mutation, C, unacceptable missense mutation, and option D, partially acceptable missense mutation. So the correct option is C, unacceptable missense mutation. Explanation is hemoglobin S is the cause of sickle cell anemia. It results from a single base substitution that changes a glutamic acid to valine, so that this change is considered unacceptable because it significantly alters the structure and function of the hemoglobin protein, leading to a severe disease phenotype. Thank you for watching my videos.